So this kind of connects, and the reason I talk about membrane potential first is so I can talk about action potential, because action potential is the triggering factor for muscle contraction. And let's face it, we're animals, okay? So that means we can move around the planet and do cool stuff. Well, how do we do that? We're going to combine our nervous system change that we've been talking about now to a physical protein-based change that's actually going to make contraction happen. And I cover muscle early on in the course because in the sister class in anatomy, you're going to learn a few muscles, right, if you haven't already. And you're going to, if you didn't learn this in your undergrad experience, you will get to, which is origins and insertions. Right? Now, not to worry, no origins and insertions in my class. All right, that's in Sean's class. But muscle contraction is shortening the distance between the origin and the insertion. It is the fundamental of human motion. It is how we move around the planet. We squeeze one end and that moves a bone, right? So we're going to start putting those pieces together here today of how does the muscular system accomplish that moving of the bones that moves us around and gets us to do cool things. All right, so muscles, fibers, sarcomeres, and components. I like to think of the muscular system as bundles of bundles of bundles, okay? And let me show you what I mean. So if you take an individual muscle, a muscle is an organ, okay, proper organ. Well, if we look inside a muscle, what we see that a muscle is a bundle of fascicles, okay? A fasciculus, that is the singular, right? So one fasciculus, many fascicles. A fasciculus is a bundle of myofibrils, or muscle fibers, synonymous with muscle cells. Remember, muscle cells are kind of weird cells because they start out as lots of smaller cells that join into these big supercells, right? It's why myofibrils have many, many nuclei, for example. It's because they started life as many cells and they kind of combine together. So a fasciculus is a bundle of myofibrils. Myofibril, in turn, is a bundle of sarcomere chains, right? So, not myofibrils. What do you call a muscle cell? The other name for it. Anybody know? Sarcomere. No, that's lower. Oh, it's gone. Anyway, muscle, bundle of fascicles, fasciculus, bundle of fibers. Fibers, a bundle of sarcomere chains, right? Those sarcomere chains called myofibrils. There's another name for a muscle cell, and I mistook myofibrils for that. But anyway. What's a sarcomere? Okay, so now we're down into molecules. Okay, the sarcomere is an arrangement of protein filaments. There are thick filaments that are made up of a protein called myosin, and there are thin filaments made of a protein called actin. It's the interaction of the actin and myosin filaments that cause sarcomeres to shorten, which causes fibers to shorten, which causes fascicles to shorten, which causes muscles to shorten. See, so it's all connected together. And we're going to sort of build up these pieces as we go. All right. So enter the concept of functional unit. In our talk about physiology, we're going to talk about a number of functional units. A functional unit is the smallest unit of an organ or an organ system that accomplishes the goal or the purpose of that organ. All right, what is the purpose of a muscle cell? To contract, right? Okay, so what is the smallest bit of a muscle cell that accomplishes that goal? The sarcomere, all right? So the sarcomere is the fundamental unit of muscle. We'll see functional units in the kidney. The functional unit of the kidney is the nephron. In the lungs, it's the alveolus, right? In the heart, it's the cardiomyocyte, okay? So the sarcomere is this arrangement of thick and thin filaments. There is a tiny bit of anatomy. We could spend a whole week talking about the sarcomere, but it has very little clinical relevance, right? All I want you to really have a sense of is how and why do muscles contract the way they do. But I do want you to know a little bit of structure. All right, so we have what's called the A band. The A band is the part of the sarcomere that includes the thick filaments. All right. The A band got discovered because when we look at this structure under the microscope, the thick filaments are very dark and the thin filaments are quite a bit lighter. So when we look at the sarcomere, we have a dark band here, then a light band, then a dark band here, then a light band, and so on. Okay? So the A band is the area that has thick filaments. Thick filaments contain myosin. Myosin is the workhorse of muscle. It's what's actually going to make movement happen, and force generated is going to be at myosin. All right, the Z disc is right along here. It's this squiggly line. The Z disc is a collection of filaments that bind actin molecules together. All right? The I bands sit on both sides of the Z disc, and the I bands are the region where there's only actin. Do you see that? There's no myosin in this section. So under the microscope, the Z bands are light and the A bands are dark. All right? So the function of this system, if we sort of zoom in so we can see some of the protein structure. All right, so here's our Z disc. Here's our I band, right, would be from here over or from here over, all right? And then our A band is the thick filaments. You'll notice that the thick filaments, I thought I had a better picture of myosin. Okay, this is what myosin looks like. It looks like a golf club. It has a golf club sort of shape to it. Well, the thick filaments grab hold of the thin filaments and pull them inward, all right, towards the center of the A band, which incidentally is called the M line, which isn't on my list. But so it means that this structure shortens in the right conditions, and we'll talk about what those conditions are. The myosin thick filaments pull on the actin thin filaments and pull the Z discs together, closer and closer together. That causes the whole sarcomere structure to get smaller in this direction, in the left and right, right? That's muscle contraction. If we do that to a bunch of sarcomeres in a bunch of myofibrils in a bunch of fibers, well, we get a shortening of the muscle or a development of tension in that muscle. So it means, and this is an important point about muscle, hopefully one you've learned before, muscle fibers can only pull. They can only contract. They can only get shorter. They cannot push on anything. It's why when we look at the anatomy of the human, we see that we have muscles on both sides of almost every joint, because that's the only way that joint can move in both directions. You need a muscle to pull in order to have anything happen. So you have one on the top that pulls, and you have one on the bottom that pulls, and that moves the bone in the two directions, let's say if it's an elbow, okay? Now, there is a tiny, tiny little bit of a push inside the sarcomere. It is not enough to move any bones around, but it is enough to sort of spread the sarcomere back out during muscular relaxation. That protein is called titin, T-I-T-I-N. It's this guy right here. It gets its name because it's a ginormous protein. Okay, so if you remember your mythology, right, the Titans were the ancient gods that were like big and nasty and mean, right? Well, they named this protein Titan because it's this big, massive protein. It's like substantially bigger than practically all their proteins in the body. It has a bit of a spring effect that pushes the Z discs Z discs apart when the muscle is relaxing. But remember, it's not enough to move any bones, okay? It's just enough to put the sarcomere in a more optimal position for contraction. So that's tighten right here. All right, so this is my sarcomere anatomy that I want you to know, okay? Don't get into more detail than this, because like I said, there's a lot known about muscle and how it works. Thanks in large part, for real, to the Muscular Dystrophy Association. They have done just a massive amount of research in how muscle works in order to understand that disease better. So much of what we know about muscle contraction was funded by those folks. Okay, so let's talk about how does the sarcomere actually contract? How are the Z discs pulled together? Okay, so here's our thick filament. Here's our myosin molecules looking like golf clubs, right? And the myosin heads, this is the head part, are going to bind to active sites, to specific binding sites on the actin filaments. When that binding occurs, this head is going to change shape and is going to pull this actin molecule this way and this way, right? Now, one of the tricks to muscle contraction, we need a muscle to contract when it's needed, but not be contracting when it is not needed, right? Otherwise, we would just be stiff all the time, and that would be no good. So we have to be able to turn this contraction system on and off. The way that we do that is through tropomyosin and troponin's relationship to the active binding sites on actin. All right, it goes a little like this. When calcium is present around the sarcomere, calcium binds to troponin, which is another protein complex. When that binding occurs, troponin takes tropomyosin, which is a long filament kind of a protein that's wound around actin. Calcium binds to troponin. Troponin then moves tropomyosin. It kind of unwinds tropomyosin a little. And that exposes the binding sites for myosin on the actin filament. That's what we need to have happen in order for muscle contraction to occur. So it means that the trigger for muscle contraction, for sarcomere contraction, for myosin and actin's interaction, that trigger is my favorite ion, right? Calcium. Okay, so when calcium is present, troponin moves tropomyosin, that exposes the binding sites. Now myosin can bind to actin and we can make contraction happen. All right? Now, using ATP, lots and lots of ATP, these myosin heads go from their inactivated or cocked position, like we see here, and they ratchet forward. Well, if you do a little imagination, if this head were to ratchet this way, it's going to tend to pull this filament that way, right? It's kind of like a tug of war. You grab the rope and you pull, right? That's kind of what the myosin heads do. They grab hold of actin and they give it a tug. They pull on it, causing the actin filaments to be pulled together and the sarcomere to shorten. Okay, so the A&P Flicks people do a much better job drawing than I do.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a crossbridge. Also note that the myosin head must be activated before a crossbridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A crossbridge cycle may be divided into 4 steps. Step 1. Crossbridge formation. The activated myosin head binds to actin, forming a crossbridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Crossbridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the crossbridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
So sarcomeres contract, get smaller. That creates either shortening of the muscle, i.e. movement, or tension development. So let's talk about muscle in action a little bit. All right, so the easiest to understand is a moving bone, right? So the muscle shortens, pulls on a bone, that joint angle reduces. Well, the other thing that a muscle can do is generate tension. So the classic example of this is, put your arm out, put a heavy book on it, right? Your arm isn't moving, but are you spending energy on muscle contraction? Yes, you are, right? Okay, so that is the other way that muscle works to move us or keep us in a posture, is by providing tension that eliminates change that would otherwise happen, okay? So the interaction of actin and myosin are what create contraction. But the sarcomere changes size, right? It does in fact get shorter. When bones are moving, sarcomeres are getting shorter. So is there a relationship between muscle length, sarcomere length, and strength? And there absolutely is. Okay, so let's look at B and C. B and C, this is the physiologic range of motion of a muscle, okay? So at B, we have a sarcomere that is compressed, like it's contracted, but we're not getting a significant amount of overlap in anything, right? In point number C, here we have a sarcomere that's kind of stretched out pretty well. Now, let's go to the extreme ones. Now, what about D? In D, this muscle is stretched to such an extent that the thin filaments have been pulled away from the thick filaments. Can that muscle contract? No, because myosin can't reach actin to make that contraction happen. So that what we see is as a muscle is stretched past an optimal point, its strength starts to fall off. It gets less and less able to contract the more stretched it is. And it's fully explainable by the anatomy of the sarcomere. What's happening is there's less and less myosin and actin interaction. There's less and less overlap. That's where the strength comes from. So as we stretch a muscle out, it becomes weaker and weaker. Now, people always ask, so does that mean we shouldn't stretch before exercise? No. This is like muscle that's been taken out of the organism and is stretched beyond its anatomic, okay? It's really to point out that the overlap of filaments is where the strength comes from, okay? Now, what about over here in A? A is an example of a muscle that's been all squished together. It is too contracted. So here in A, what we see is the thin filaments are actually overlapping. So there's less interaction between myosin and actin because a bit of the filament is actually overlapping itself and not anywhere near myosin because it's in the center point, the M line. So we see the same thing happen. A muscle gets weaker as its length becomes short enough that we don't get optimal interaction. Okay? So that's that bit. The other bit that we see is this is whole muscles. Now this is just sarcomeres, but now we're going to look at whole muscles. Here we see that at the normal length of a muscle, it has its maximum tension, or maximum tension is just another name for muscular strength or contraction force, right? And as the muscle gets shorter and shorter, its ability to create tension goes down. Similarly, we see that as it gets longer and longer, it starts to go down. Now, what about this little bit here where it starts to go back up? That is a different thing happening. That is muscle has an intrinsic, it holds itself together. So if you try to stretch a muscle too far, it starts to resist that stretching. Okay, so the tension that you see in the little upstroke there has nothing to do with the sarcomere. It has to do with the elasticity of muscle, and it doesn't want to be pulled apart, right? Okay. So length and strength have this relationship. In the heart, we see this relationship play a really significant role, and in what we call the Frank-Starling relationship. And it goes a little like this. The heart is a little too small in the normal like in our normal everyday lives. The heart is a little less stretched than its optimal. But that means that when we stress the heart, it stretches a little and then can work even better than it does when it's at the normal condition. We'll talk about that as the Frank-Starling relationship in a week or two. All right. Okay, so kind of filling in on the what I was talking about with the book versus movement. We have isotonic versus isometric contraction. This has a tendency to confuse students, so I always just touch on it briefly. In an isotonic contraction, I like to take words apart and show you what they mean, okay, so that they're not foreign. Iso is same, tonic is force or contraction, okay? So isotonic, what we're saying is the same force, isotonic, is being applied. All right, so if we apply the same force to a muscle, it's going to shorten, right? So when the muscle contracts, it gets smaller. When the muscle relaxes, it gets longer. That's the typical moving of the bone. Now, isometric, iso same, metric measure, length, same length is what isometric means. That is the book on the extended arm. Nothing is moving, but the muscle is staying the same length despite forces trying to push it around, okay? So that's what we call an isometric contraction, where tension is present, but motion is not occurring. Isometrics happen all the time. That's what your core is for, right? It's to keep your posture. It's to keep your scapula where they belong and where your and the pelvis where it belongs. So we actually spend a lot of our metabolic energy on isometrics every day. Okay. Then the last thing I'm going to talk about is that not all muscles behave the same in terms of how they contract. Some muscles contract relatively slowly but with a lot of strength. Other muscles contract very quickly but with much less strength. Okay, so for example, the eye muscles, the muscles that move your eyeballs around, all right, they're very fast, like they contract very quickly, but they don't have a lot of strength because, let's face it, all they're moving is an eyeball around, okay? Now, compare that to the soleus. You know, the soleus is your primary walking muscle. Well, it's kind of slow to contract because it's not the jumping muscle, it's the walking muscle, and walking doesn't require fast contraction. But it is very strong, right? So, depending on what the muscle's job is in the body, it could have fast contraction or slow contraction. We talk about this typically as fast twitch and slow twitch. And it's a difference in how the sarcomere responds to the contraction signal. All right? Now, know that most muscles have a mixture of fast and slow twitch, but one will predominate over the other. You know, so like the eye muscle, classic fast twitch. It's quick, responsive, but not very strong. Slow twitch, soleus, your gluteus maximus, all your glutes are of the slow twitch, high strength variety. The slowest twitch that we have are our postural muscles. I'm assuming in anatomy, did you do the back already? Yeah, we usually start you with the back. So you've seen the paraspinal muscles. The paraspinal muscles are the slowest twitches there are. It's why they look a little different than like a bicep muscle does, because they're all slow twitch fibers. All right, so if we zoom out a little and we look at the muscle as an organ, well, the individual fibers in that muscle have an arrangement with the brain, okay? And that arrangement is called a motor unit. So a motor unit is all of the muscle fibers that are controlled by a single motor neuron. All right, so that statement implies that one motor neuron controls many muscle fibers, and that is correct. All right? Now, the variance here, the difference is that some muscles have a lot of motor units. Some muscles have only a few motor units. Okay, that's one variable. Another variable is some muscles are really big, some muscles are really small. Okay, so we have the total number of fibers in a muscle also varies. Okay, so if we have a large muscle, like say, one of your quadricep muscles or the gluteus maximus, well, there we've got a lot of muscle fibers because we have to do a lot of work, right? But we have relatively few motor units. In other words, each motor neuron is controlling a lot of muscle fibers. Now, that's fine in a large muscle because its primary role is to do bulk work, move the body up a flight of steps, for example. Now, if we look at a different kind of muscle, like the muscle in the eye, okay, the muscle in the eye is very small, but it has a lot of motor units. In other words, there are a lot of motor neurons that control that small muscle in the eye. Well, what is the purpose of that? It allows very fine control. Okay, the more motor units there are in a muscle, the better the brain can control that muscle. All right, so another example is the muscles that flex your fingers. okay? Many of you are typing or writing at the moment. Well, that's a fine motor, like the brain has to have very careful control of the muscles in order to do that. So those muscles in your forearm, again, have lots of motor units, even though they're not particularly large muscles, okay? So motor units allow control by the brain of the muscle. More motor units, more control. Now, the way this sort of looks, motor units are all mixed up. So in a given part of muscle, your motor unit 1 has multiple fibers spread out, 2 multiple fibers spread out, and so on. So they're distributed inside the muscle. Now, the benefit of that is it means that we can use different motor units over time. So I gave the example the other day of holding a heavy book on your extended arm, right? Well, one of the challenges with that is that muscle has to stay contracted for a long period of time. Motor units, motor cells, our muscle cells, get tired, right? They run out of ATP, they need to rejuvenate. Well, the motor unit system allows a single muscle to break up the work in different motor units. So for example, in our holding the heavy book analogy, motor unit 1 may do the work at first, and then when it starts to fatigue, motor unit 2 picks up while motor unit 1 goes down, and so on. And then by the time we get to, say, motor unit 3, motor unit 1 is rested again, and it can start up as well. So the motor unit system allows different muscle fibers to be active at different times so that we can sustain contraction for long periods of time. And we do that. Our back muscles, for example, if you're not lying flat in bed, they are contracting. They are holding the rest of you up. And they take advantage of this as well. Now, you can imagine that those muscles in your back, you don't have very fine control of those, right? So it's not like they have lots and lots of motor units, but they have enough motor units where they can do the taking turns thing, okay? So that's the motor unit. Now, when we talk about innervation, this nerve innervates this muscle, we never get into the detail of which motor unit in which muscle, okay? But just know that, like in the cadaver lab, the anatomy lab, when you're looking at a nerve, you're looking at lots of different axons, right? And each of those axons is going to control a different motor unit when it gets to the muscle, okay? All right, so that's motor units. I already talked about this. So few fibers, more control, lots of fibers, more strength, less control, and then a crazy example. It's why we write with our hands, not our feet, right? Because our leg muscles don't have very fine control. Now, yes, you can go on YouTube and find people, writing with their feet, but that is a learned skill, not something that most of us can do. All right. Muscle uses a ton of ATP. It's very energy hungry. So we have to talk a little bit about the fuel system that allows muscular contraction. Well, the first fuel that muscle use is stored ATP. All right, muscles do keep a fairly large amount of ATP kind of stored up when they're relaxing. Now the issue is contraction consumes so much ATP that storage only lasts about 1 to 2 seconds. In other words, enough for a brief contraction and that's it, okay? After that is used up, the muscle moves to its creatine phosphate system. Creatinine and creatine, they're unfortunately named, all right? They are not the same thing at all, all right? Creatinine is a measure of kidney function. Creatine is a component of muscle, all right? So creatine phosphate or phosphocreatine, this is a molecule that has a high energy phosphate bond that can be very quickly moved to another molecule, all right? So when the muscle is at rest, it creates a bunch of creatine phosphate. When its ATP runs out, it has a lot of ADP around, right? All right, so then the magic comes. The ADP steals the phosphate from the creatine phosphate and regenerates ATP, all without needing to go through our friend the glycolysis cycle, right, and the oxidative phosphorylation. So creatine phosphate works like a battery for ATP. When the ATP starts to run out, in other words, when ADP levels start to rise, creatine phosphate can very quickly put back the ATP. Now, this isn't a sustainable process though, right? Because at some point, the creatine phosphate is just going to be creatine. That's all that's going to be left. Now it can't do its recharging trick anymore. So we go to the next level of energy production, glycogen. We talked about glycogen on our first day together. Glycogen is long branching chains of glucose, right? Glucose is a preferred fuel in lots of places, but one of the nice things about glucose in muscle is we get a tiny bit of ATP out of glycolysis, and glycolysis does not require oxygen. So a muscle that is in an anaerobic state, in other words, it's doing more work than it has oxygen to supply it, we can use glycolysis to very quickly create ATP. Now, the downside to this is that it isn't very efficient, right? Glycolysis only produces a tiny bit of ATP compared to oxidative phosphorylation. All right, so that's level 3. And it means that muscle has a lot of glycogen in it. At least healthy muscle that is well exercised, right? Okay, so then the fourth layer, oxidative metabolism. So this is your classic biochem stuff, right? Where you feed glycolysis products into the oxidative phosphorylation cycle and you get oodles of ATP out. Now the downside with this last one, this is the most powerful of the energy sources. This can go on as long as the body can supply fuel to the muscle and sufficient oxygen. The downside is it's slow. Oxidative phosphorylation takes time. It's very efficient, but it's not very fast. So it works best in a muscle that is not at its extreme in terms of activity. When you exercise or have had exercise classes, they talk about anaerobic versus aerobic muscular activity. This is the difference. Aerobic muscular activity is keeping the muscle at an activity level that oxidative metabolism can supply the ATP, right? In other words, you're not sprinting, you're jogging. Because at a sprint, you're not going to have enough oxygen fast enough to create the ATP you need. At a jog, you will. Okay? So fuel sources of muscle. Oh, yay, Coot. Don't be on like Snapchat in my class. Not that I'll go look, promise. Okay, where's the Z disk? Oh, that's an easy enough question. Good, okay, that's in the middle of the I band is the Z disk. Okay, this is what I call a level 1 question. In other words, it's asking you for one and only one fact. You have to know exactly one thing. Do not expect level 1 questions on your exams in this program, okay? That's one of the things that changes as we go from undergrad to grad, right? We are going to ask you to apply what you know, not simply know things, all right? I'll try to give you an example of a level 2 question as we go here today. That was a quick one. All right, what is required for myosin to bind to actin? All right, good. Yeah, that's all of those. Why? because you got to have all of them. The triggering factor is the first one. Troponin binds to calcium, or calcium binds to troponin is probably the more proper way to say it. That causes tropomyosin to move. That exposes the binding sites on actin, and then actin and myosin, if they're close to each other, will bind. All right. A muscle has many motor units, each with a small number of fibers. So what's true? All right, good. So this is kind of a level 2 question because I've given you some facts, but I'm asking you to draw a conclusion from those facts, right? So if a muscle has many motor units, that means it's going to have fine control. And if it has only a small number of fibers in each of those units, it's probably going to be a small muscle, right? So small muscle with fine control. To give you a counterpoint, large muscle would have few motor units, each with a lot of muscle fibers. Okay, so that's sort of the flip side. All right. Last one. Which of these is not a primary fuel of muscle contraction? Yeah, protein. If you've got a muscle that's burning protein, you have a problem, right? And technically speaking, muscle doesn't have the metabolic machinery to burn protein anyway. You'd have to run it through the liver first. So creatine phosphate is that energy battery. Glycogen comes next because glycogen becomes glucose. Through glycolysis, we get ATP. And then fatty acids. I didn't make a big enough deal about this a second ago. We got a lot of fuel sources circulating in the blood, right? Glucose is one of them, fatty acids is one of them. Muscle actually prefers to use fatty acids over carbohydrates because fatty acids contain a lot more energy per gram. And muscle being hungry, so to speak, it prefers that high energy molecule as its fuel source. All right, who's our winner? I've seen a social lobster or two. It's kind of a joke. Not a good one, apparently. All right, we'll get rid of that. Onward. Okay. So now we've got a muscle contracting. Well, the next bit that we need is we need to connect that muscle contraction to some kind of nervous system control, right? So that we can have a brain controlling that muscular contraction instead of just randomness. Okay, so enter the neuromuscular junction, also called the NMJ, for those who don't like to write out big words, neuromuscular junction. This is where the motor neuron coming from the spinal cord ends at the surface of a muscle. Okay, so nervous to muscular system junction. This is a modified synapse. Remember, a synapse is where two neurons come together. One neuron sends a neurotransmitter across, makes something happen. All right, well, here we don't have two neurons. We have a neuron in a muscle cell. Okay, so we have our axon coming down. It ends in this kind of foot that then is on the membrane of a myofibril. Okay? Well, when this axon fires, in other words, when the lower motor neuron is activated, this axon is going to release acetylcholine from its terminals. It's going to release that acetylcholine in response to calcium entering the cell again. There's calcium again, right? So calcium enters the cell. That triggers the exocytosis of pre-made neurotransmitter. That neurotransmitter crosses the cleft, the little gap in between, and it activates channels that look like this on the muscle cell membrane. Okay, so this is a chemically gated sodium channel. That ought to sound familiar because we talked about them just the other day, all right? We're just making it specific to acetylcholine. So it's normally closed when acetylcholine binds to this channel. The channel opens and allows sodium to enter. Because of the concentration gradient, sodium's going to enter quite quickly, right? And is going to trigger a change in membrane potential, right? This should all sound very familiar. So the acetylcholine triggering a membrane potential change, that membrane potential change is then conducted across the muscle cell membrane and triggers the release of calcium, which then causes the muscle to contract. All right? So if we zoom in and look at just one area of this, we see that inside the neuron, we have oodles of mitochondria and oodles of vesicles, all containing acetylcholine. What this should tell you is this is a very responsive and fast structure, right? The energy is being made nearby, and we got a lot of pre-made molecule. Remember in physiology, anytime you have to make a thing in a cell, that takes time, okay? So if you want a thing to happen in milliseconds, it needs to be pre-made, okay, which is what we have at the axon terminal here. Okay, did I cover all my things here? Yeah. Another movie. Turn off my lights for me. I already checked this one, haha, so I know it will work.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as the sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so now we've got our action potential onto the sarcolemma. We've gone from nervous system to muscular system. So now what we have is we have an action potential that's moving along the surface of the muscle cell, moving along the sarcolemma. Well, we need to connect that voltage change, that membrane potential change, to calcium release, because we've already learned we need calcium for the muscle to contract, right? So the way that that happens is through the T-tubule system, all right? So if we look at this, this is a broken up muscle cell, and we have, so this is the surface, this is the sarcolemma. Dipping down into the cell and around the myofibrils is a system called the transverse tubules and the terminal cisternae. All right, these are modified endoplasmic reticulum. So if you remember, ER is basically just a membrane organ, right? It's a structure, a bag of membrane. Well, in the muscle cell, endoplasmic reticulum has modified into sarcoplasmic reticulum, which is basically a holding zone for calcium, all right? So when the muscle cell is at rest, the SR, sarcoplasmic reticulum, the SR, is chock full of calcium. When the action potential arrives down the T tubule and into this area, that voltage change causes calcium channels on the SR to open. Now, in most cells, most of the time, calcium levels are kept very, very low. You can always remember that, because if they were high, things would be happening, right? So the calcium level is kept very low. So when these voltage-gated calcium channels open, whoosh, we get a bunch of calcium into this area around the sarcomere. That calcium binds to troponin, which moves tropomyosin. You know the rest of the story, right? We call this process excitation, contraction, coupling. Excitation is the nervous system has activated the muscle cell, okay, so we have a voltage change, connecting it to contraction, actually getting the muscle to shorten. The bit that connects those two is calcium. When calcium gets released from the SR, we get contraction. All right, so how does that happen? Well, here's our sarcolemma. This is our action potential moving down the T tubules, right? Well, what happens down here that actually causes calcium to be released? Well, there is a receptor in the T tubule called a DHP receptor. That DHP receptor kind of has a shape like this. It has a cork on one end and a receptor on the other. When the action potential moves down the T tubule, DHP pulls the cork on this calcium channel right here. That then causes the calcium that's built up in the SR to leave and cause contraction. Now, when the muscle cell, when the sarcolemma repolarizes, the DHP receptor puts the cork back in. Now calcium is once again sequestered in the SR, and calcium channels, oodles and oodles of calcium pumps, pump the calcium from the cell cytoplasm back into the SR so that the muscle can then relax. Now, it's important to know, as is true with everything in physiology, this takes time, which means that a muscle can't go from contracting to relaxing instantly, right? We have to have some time for the calcium to be pumped back into the SR. As the calcium concentration goes down around the sarcomere, relaxation occurs, right? So we have kind of a slow relaxation and a slow contraction, relatively speaking, because of these things moving around. Relaxation, as you might imagine, is a little slower than contraction is. Because contraction can happen real fast because calcium just has to go down its diffusion gradient. Relaxation happens a little slower because calcium has to be pumped back in to the SR. Yes?
Is this kind of like a reason for like after you exercise and stuff for your muscles to remain kind of tight and flexed for a little bit after you've done exercise?
It can be. You can have a general calcium excess around. Because when you contract a muscle a lot, not all the calcium ends up back where it should be. So you can end up with increased muscle tone afterward. Yep. All right. Now the other bit is this calcium pump requires ATP. Yet another reason why muscle cells are hungry, right? A lot of the things they do require ATP. All right, last movie, another light show. Doing great with the lights, thank you. They can just make better pictures than me. So I like the movies.
A single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with the calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape.
That's
This
DHP.
shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
All right, so that's excitation, contraction, coupling. You can hit my lights for me again. So now you know the whole story, right? So you go from brain to neuromuscular junction and motor end plate, and then into the muscle, and now you've got a contraction to occur. And then relaxation is basically the opposite of contraction. So when there is no signal from the nervous system, then the muscle relaxes. The DHP receptor closes the calcium channel, and the calcium pumps remove all the calcium from around the sarcomere, and we get relaxation then that occurs. Okay? So that is skeletal muscle. Now what we're about to talk about here is smooth muscle. All right, so we have three different kinds of muscle in the body: skeletal, smooth, and cardiac. Cardiac muscle is kind of a blend between the other two, as we'll start talking about tomorrow. We have to talk just a little bit about smooth muscle because there's lots of it in the body, okay? We find smooth muscle in all of our arteries. We find smooth muscle to a lesser extent in our veins. Our entire digestive tract is made of smooth muscle. The uterus is made of smooth muscle. So we have basically two different kinds. We have multi-unit smooth muscle, and then we have the more typical or unitary smooth muscle. Multi-unit smooth muscle is kind of somewhere between regular smooth muscle and skeletal muscle. So it has discrete, separate fibers. There is nervous system stimulation that causes the contraction of multi-unit smooth muscle. And it has relatively fine control compared to unitary muscle. Now, the true issue, though, is it isn't found in very many places. Okay, there's like 3 places in the body where we have these. In the eye, ciliary muscle that controls the shape of your lens, the iris that changes the pupil size, and then the piloerector muscles, which are your goosebump muscles. Okay, so those are the only places we have multi-unit smooth muscle, so we can kind of effectively not think about it. For unitary muscle, we also call it syncytial smooth muscle. That's what I tend to call it, is syncytial muscle. A syncytium is just a group of things working together. All right, so a syncytial smooth muscle, you've got lots of different muscle cells, but because of how they're connected, they tend to do things at the same time. So they kind of help each other. So we get many different cells that work as a group. Now, important to know in smooth muscle that most contraction is not controlled by neurons. Instead, the contraction is controlled by the muscle itself in response to chemical or environmental signals. All right? So like when we talk about the GI tract, we're going to talk about the fact that the GI tract has a brain, but that brain doesn't control things very precisely like the brain in your head does. So in smooth muscle, we have these groups of cells that are all connected together, and in response to chemical or environmental stimuli, they contract together. All right, so what is similar and different between smooth and skeletal? Well, we still have actin and myosin. The single biggest difference between smooth muscle and skeletal muscle is how actin and myosin are arranged. In skeletal muscle, actin and myosin are arranged in those very elegant sarcomeres. So we get contraction in just one direction, right? In smooth muscle, the arrangement of actin and myosin is more like a web, right? So instead of getting contraction in one direction, we get contraction in all directions at once. So it's kind of like a rubber sheet, right? You know, you can stretch it and it will contract in lots of different directions. That's kind of how smooth muscle works. Another big difference is actin and myosin still make cross bridges. Those cross bridges are still driven by calcium entry, okay? But they're much slower. Smooth muscle is not fast. It is instead slow and strong. So for a given gram of smooth muscle is much stronger, in other words, able to generate more tension over a longer period of time than the same amount of skeletal muscle is. So it's slower but stronger, okay? So the maximum force generated per unit volume is higher. Another thing that smooth muscle has that skeletal muscle does not is a latch mechanism. So in skeletal muscle, contraction always requires myosin heads pulling, right? The tug of war, the constant tug of war. Smooth muscle has the ability to contract and hold without spending a lot of energy. It has a kind of latch that keeps actin and myosin bound together, but without contraction occurring. It's good at holding tension. This plays a significant role in our arteries, right? Where they have to hold tension all day long, day after day after day, right? So this helps them to do that with less energy. And then the length-tension relationship is very different. We saw some graphs yesterday about if you stretch a muscle too long, it gets weak. If you squish a muscle too short, it gets weak. Smooth muscle has its consistent tension development over its entire range of lengths. So like in the GI tract, for example, when you have food passing through your intestine, it can stretch out pretty good. That muscle doesn't get any weaker as it stretches out. It keeps its strength whether it's this size or this size, okay? So it has sort of a better relationship between those two things. But ultimately, like any muscle, it needs to contract when we need it to contract and relax when we need it to relax. Oh, can you close my door so I don't interrupt the art students' creative processes? Okay. So how do we get smooth muscle to contract? Well, still calcium. See why calcium's a favorite of mine? It's in everything. Okay, so but the main difference is where the calcium comes from. Okay, so yesterday we talked about excitation, contraction, coupling. How an action potential from the nervous system leads to an action potential in the muscle cell, which causes the release of calcium and the myosin-actin cross bridges. All right? Well, in smooth muscle, we don't have T-tubules, we don't have a neuromuscular junction, we don't have sarcomeres. So how does the calcium make anything happen? All right, well, it's going to come in from the outside. So the extracellular fluid always has a fair bit of calcium present relative to the inside of the cell. That was in that early table I showed you with the differences. So if we open a calcium channel on the membrane, the cellular membrane now, not this SR, there is no SR in smooth muscle. Well, there's not a very mature SR anyway in smooth muscle. If we open the calcium channel, calcium's going to come rushing in. Now, where does it go? Well, in smooth muscle, the actin and myosin filaments are not arranged in sarcomeres. They're arranged more in kind of a loose web configuration. but they still respond to calcium. When calcium is present in the smooth muscle cell, that smooth muscle cell is going to be contracting. Now, our mechanism is quite a bit different. There is no troponin and there is no tropomyosin in smooth muscle. Instead, what we have in place of that is this molecule called calmodulin. That's CAM, is calmodulin. Calmodulin, when it binds to calcium, it transforms this MLCK into its activated form. That MLCK then catalyzes the breakdown of ATP into ADP and P, and we get a phosphorylated myosin. That phosphorylated myosin, not surprisingly, activated myosin, is going to bind with actin, and then those two are going to have contraction together, right? The myosin is going to pull on the actin, and that cell is going to get smaller in all directions, not in just one direction, right? So most of the calcium in this process comes from the extracellular fluid. Smooth muscle does have a very poorly evolved sarcoplasmic reticulum that does sequester some calcium. And the trigger for the opening of the calcium channels on the SR is the entry of calcium from the extracellular fluid. So there is no DHP receptor in smooth muscle. Instead, the small amount of calcium from the SR is released when calcium enters from the outside. The technical way of putting that is when the calcium concentration in the cytosol goes up, i.e. from the outside, the calcium channels on the SR open to further increase that calcium concentration. It's another example of a positive feedback mechanism. All right. Well, how about relaxation? Same general idea. In order to get a smooth muscle to relax, we have to take the calcium away, because calcium is what causes it to contract. But once again, the mechanism and the locations are a little bit different. Okay, so here we get rid of the calcium by pumping calcium out of the cell. All right, so here's a calcium pump on the surface. We also pump some calcium into that poorly evolved sarcoplasmic reticulum, right? So we sequester some there. And then finally, there's a sodium calcium pump that plays a role in the smooth muscle cell as well. This is a countertransport, so this doesn't require ATP. What we have here is we have sodium falling down, its diffusion gradient is pushing calcium up its. Right? So this is up the concentration gradient, sodium is down. Okay, so if we're removing calcium, that means that our calmodulin is going to lose its calcium that's attached. Once that calmodulin loses its calcium, that, number one, inhibits the MLCK enzyme, so phosphorylation stops, and it activates myosin phosphatase, which takes the phosphate molecules off of myosin and turns it into inactivated myosin, which then no longer binds to actin. If we end that actin-myosin binding and interaction, then that muscle relaxes, that smooth muscle relaxes. So still calcium as the trigger, but the processes are different. A couple of other things are different because of the first difference. One is smooth muscle is slower to contract. You know, a skeletal muscle can contract in a few milliseconds. Smooth muscle is slower to contract and slower to relax. But that works just fine because smooth muscle tends to be in places that depends on sort of a slow, steady contraction and a slow, steady relaxation. The best example I can give you of this is the uterus, right? During delivery, at the end of pregnancy when the baby is born, we see the uterus have contractions, right? We've all seen this on TV if we haven't seen it in real life. Well, those contractions are not like on and off, like a skeletal muscle. Instead, they sort of build, and then they relax. And the reason for that sort of slow up and down is because of this different calcium utilization system in smooth muscle. It just takes longer. It still works, but it takes longer to contract and then relax. All right. Okay, so where does the signal for calcium to enter the cell come from? Well, one of the key important differences between smooth muscle and skeletal muscle is smooth muscle is not under direct control of the nervous system. The nervous system plays a role in smooth muscle contraction, but it's more of a role of increasing the likelihood or decreasing the likelihood, not triggering a contraction or not triggering, right? So the contraction of smooth muscle, the opening of that calcium channel on the membrane, occurs because of chemical and environmental factors much more so than nervous factors, all right? Now, another difference between skeletal and smooth muscle can be inhibited as well as excited. So if the nervous system isn't causing contraction, but it's regulating it, well, how does that happen? The release of neurotransmitters from the nerves around smooth muscle causes either excitation, an increased tendency to contract, right? Or inhibition, a decreased tendency to contract. So there still is some control by the nervous system, but it's less point to point than what we see in skeletal muscle. Now, how that signal is transmitted from smooth muscle cell to smooth muscle cell differs between our two different types of smooth muscle. In unitary smooth muscle, remember I said that was by far the most common type, right? Unitary smooth muscle. The cells, the individual smooth muscle cells, are connected to each other with what's called a gap junction. Okay, so what is a gap junction? Well, let's say we have cell A and cell B, right? A gap junction is a protein that makes a kind of tube that connects A and B, right? So what that means is calcium that enters this cell, let's say, is going to have a tendency to flow into its neighboring cells because of these gap junctions that allow substances to pass. So it means that if we excite or if we activate open calcium channels on the outermost cells, that calcium will flow from cell to cell to cell and cause that whole group of cells to contract all at once, right? So this is why we call it a functional syncytium. A change in one cell is transmitted to the other cells so that they work together as a team, okay? Now, in multi-unit smooth muscle, remember only a few spots, piloerector muscles for the most part, okay? In multi-unit smooth muscle, we don't have gap junctions. Instead, our nerve fibers kind of intermingle with the smooth muscle cells and have a more direct effect on their contraction. Okay, so then we look down here and we see that one of the differences between skeletal muscle, which is what these graphs are from, and smooth muscle, is that calcium entry is prolonged. Okay, so we get what's called the plateau phase of an action potential. You know, this is what our action potential looks like on a skeletal muscle. On a smooth muscle, it looks more like this. We have a big depolarization, and then we have this kind of linger before it returns back to its baseline. This linger right here, this is calcium entering the cell. And remember that calcium entering is the trigger for contraction. Now, we will see in our cardiac chapter that very same pattern in cardiac muscle. So I'm going to sort of put a pin in that idea until we get there, okay, of exactly how we get that shape. We are going to talk more about that. But for now, just know the plateau, this bit right here, is calcium entering that cell. Okay? All right. Nice, nice. All right, question number 1. Oh, that's right, these are puzzles. Okay, so put the events at the neuromuscular junction in the correct order. Uh-oh. Now you got to like build something.
Where do you go?
You just move them around.
Oh.
Drag and drop.
Oh, okay.
And I give you more time on these.
Which made you red?
You guys figured it out? Now you're well known to be the most technologically adept generation ever.
Well, I'm not allowed to move them anymore.
Make us proud.
Here we go. I have to go.
You figuring it out?
Yeah. I don't know.
Well, keep working on it. There's 3 more. All right. So, ho 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 ho. Okay, so correct order. First thing that happens comes from the brain, right? Now, where does the brain thing come from? Your higher planning centers, right? You know, like it involves what's happening and what you're trying to do and all that. But what we get at the muscle is we get a neuron action potential coming down from a lower motor neuron. That causes the release of acetylcholine. Remember, that release is triggered by calcium entry, calcium or voltage-gated calcium channels at the end of the axon. That acetylcholine opens ligand-gated sodium channels, right? So then we get an action potential going in the muscle. And then finally, that opens muscle cell voltage-gated sodium channels. So the acetylcholine makes a membrane potential change, which then opens the voltage-gated channels that generates an action potential. Okay? See, we've talked about a lot already. Okay, next one. Oh, we have a space crane and a space pigeon on this one. Very nice. Okay. Another puzzle. Put the events of excitation, contraction, coupling in the right order. And in case you forgot, okay, that's action potential to contraction, right? All right. Oh, look at that. You couldn't do that again if you tried. 50-50. All right, that's... so some of you have some looking to do with this. All right, so first off, we start with the muscle cell action potential. That's where we just left, right? Voltage-gated sodium channels are activated on the muscle cell membrane. All right, so muscle cell action potential travels across the sarcolemma down through the T-tubules. Right? Once we get down into the T-tubules are where those DHP receptors are. All right? So the DHP receptor is activated. Remember, one end of the DHP is in the T-tubule, the other end is in the sarcoplasmic reticulum. Right? So when that DHP receptor is activated, it pulls the plug, letting calcium out of the SR. So calcium comes flooding out. That calcium binds to troponin, right? And then troponin, tropomyosin, actin, and myosin bind, right? And then we get contraction. Okay. All right, next one. Last one. So put the events of muscle contraction in the right order, in case you didn't see that coming.
wish you could take back your answer.
Oh, is it one of those, like, once you submit, you're in?
Yeah.
You're in for good?
But I'm like, no.
That's how our tests are, though, you know? All right, okay, so most of you got that one. So troponin binds to calcium, right? Where'd the calcium come from? From the SR, good. All right, troponin causes tropomyosin to move. That exposes the myosin binding sites on actin. The reason actin is called actin is because it has the active sites. Okay, that's why it's called actin. So myosin binds to, or exposes the myosin binding sites on actin. Not surprisingly, myosin binds to those binding sites. And then we get that myosin head pivot routine, right? ATP joins, the head is cocked, right, by the hydrolysis, but everything stays in place. The phosphate is released, and that causes the downstroke, and then the other bit is released, and that causes the actin and myosin to separate. So, reminder of all those steps. Okay, let's see how you did on the puzzles. Well, mystery dragon. Yay. Love dragons. That's one of my spirit animals. I have a pretty cool dragon on my wall. If you haven't been by my office, you should come see it. My daughter made it for me. All right, leave that one. And let's do a little Kahoot on smooth muscle.
Going back to back.
Yep. All right, 64377. Hey, we got a 5-digit number this time. And just as you're logging in, one of the things I like to remind students early on in the semester like this is, generally speaking, my Kahoot questions are far easier than my exam questions are, okay? Like, I don't want to give you a false sense of security. The Kahoot questions I sort of design them so if you've been paying attention, you'll be able to get them. So that's study-free learning, right? I know you wish it was all like that, but it's not. All right, here we go. Okay, smooth muscle does not have which of those items? Yeah, sarcomeres. That's what makes skeletal muscle special, right? We will see in our cardiac chapter, cardiac muscle also has sarcomeres, but they're modified. They're sort of three-dimensional sarcomeres instead of long straight lines. Smooth muscle absolutely does not. It's how it gets its name. It doesn't have any of those areas of striation where you have light and dark. But we do still have actin and myosin. They're just in a different arrangement that doesn't include the sarcomere. All right. A group of cells working together. All right, good. Syncytium. And incidentally, syncytium's a decent Scrabble word. Right? Because you have two Y's, only an I and a U, and then a whole bunch of consonants, right? So I like Scrabble. You'll hear me talk about Scrabble words too. Next, onward. What causes smooth muscle to contract? Okay, this is a good example of a best answer question. Because the calcium that's released from the sarcoplasmic reticulum, it does cause the smooth muscle to contract, But what causes it to be released is calcium entering from the ECF, right? So the better answer is the calcium coming from the outside. And if you were to follow the amount of calcium from the two places, the calcium entering from the extracellular fluid is the much more significant amount of calcium compared to what comes out of the SR. We're going to see that same pattern in the heart muscle as well, because it has sort of a weakly evolved SR as well. Okay, very good. Okay, 4 out of 5. Which of these is important to smooth muscle contraction? You guys are getting quicker. Very good. Gets calmodulin. Calmodulin is the, not surprisingly from its name, it's the modulator of contraction. It's the protein that takes the calcium input and turns it into contraction through that phosphorylation activation. All right, last one, and then we got to get back to me talking. All right, at this end of the second week, I'm feeling how about my classes? And they're all right answers. Okay, so this is more of like a survey. Which I'm looking intently at. Good, good, that's about right. All right. Nice. It's accessible. You know, you're going to work harder than you've worked in school before, but the vast majority of students, the work pays off and you're able to be successful. All right, who's our winner today? Rocky Lobster, very nice.